reduction and oxidation, right? And so we said that one of them is going to cause you to lose electrons. Which one was that? Was that oxidation or was that reduction? Oxidation, good. Remember Leo, lose electrons, you're getting oxidized or oxidation. And then we said if you gain electrons, you're reduced. Yeah, reduction, exactly. Now, it might sound a little confusing when you think about that because usually when you're reducing something, that means you're losing something, right? But remember what electrons are. They are charged negative particles. So when I'm gaining them, my charge is reducing. Does that make more sense? So my charge is becoming more negative. I'm reducing my charge. So that's why we look at it as a reduction. All right? I think this was our last slide, right? Reduce and oxidize. So the reason that this is going to be important is because it's going to come into play when we go through and start talking about metabolism, which is coming up in our next few slides. And metabolism basically is going to be a stepwise process of oxidations and reductions. So we're going to be stealing electrons from something, putting those electrons somewhere else, and then adding those electrons to something else at the end. Yes, sir? Oh, yeah, I don't know why it's doing that. Let's see. Let me escape this for a second. Sorry, guys, hold on. Let's see if I can do my display settings here. Is that doing it? No? Oh, how funny. Okay, so it's either they see it or you guys see it. How funny. It's not letting me choose one or the other. All right, we'll start it again. Hold on. And then I'll switch it back. This one's good? All right. All right, so I know you guys at home are seeing a split screen. It's weird. It's either letting me do a split screen or a non-split screen, so we're going to go with this. Okay, so each step of the process then is going to require its own enzymes. Remember, enzymes are proteins. They help processes occur. We're going to talk about that today too. All of the steps are highly regulated. What I mean by that is you're not going to have metabolism just occur willy-nilly. It's going to be highly regulated in the body because it's a big process, and we want to make sure that we're spending our time and energy wisely. When we're done with metabolism, the goal is to have a new final product. So again, it's like rearranging our Legos to get a final product. All right, so remember, when a molecule is reduced, we said that that means that it is going to be gaining electrons, right? Gaining electrons. We said oxidation means it's going to be losing electrons. And again, you can think of the acronym LEO-GER. One minimum requirement for cell growth? Well, one minimum would be that we have to have metabolism. And with metabolism, we're getting proteins, carbohydrates, DNA, and then our nucleic acids, we could just say, and then lipids. So any of those would be one for growth. And then what is meant by metabolism? It's a stepwise sequence that is going to change one product to another to sustain life. So metabolism is a stepwise sequence, changing one product to another to sustain life. And so we're going to be hitting on that as we go through our processes. And we're going to find that some organisms can do different types of metabolism than others. So a stepwise process for converting one product to another to sustain life is metabolism. So let's start with those enzymes, because we mentioned enzymes before. We just said they were proteins. I said we'll talk about them later. Well, today's the day. Today's the day we get to talk about them. So enzymes are going to work in our bodies because they're going to help us increase reaction rates. What that means is that they're going to help bring in chemicals that are needed to transform another product or another chemical that's present. So the thing about an enzyme is it will come in, it will act on a molecule, it'll change it, and then it will release it. And then that molecule can go to the next step of the process. Now, the thing to keep in mind with enzymes, when they do their work, they're not changed. So an enzyme will grab something, it will alter it, and then release it, but the enzyme itself is not changed. The best way to think of an enzyme is to think of something like your door key. Okay, you can talk about either your car key or your home key. All right, so let's talk about the home key. We have our keys, and our keys are specific, right? You have a car key, you have a home key. Will they work interchangeably? No, they're specific. That's how enzymes are. Each enzyme is unique for one process. All right, I go home and I put my key into the lock. The lock would basically be like the little substrate that I'm working with. I turn the lock and I pull my key out. I have changed what's happened with that lock, but have I changed my key? Has it deformed? Has it changed? No. And can I use it again and again? Yeah. That's what an enzyme is. So it's something that we can use to unlock something or change something. We can use it again and again and again. But the idea is just like our home key will only work in a certain lock. And that's the same thing that happens with our enzymes. So our enzymes then are going to react with a substrate. A substrate just means anything that fits with the enzyme. And then what's going to happen is they're going to release it changed. That's going to be our product. So at home, if we do our lock and key, we could think of our lock being like their little substrate and our key being our enzyme. When we turn it, the lock is turned, and that would be our product, is the turn lock. All right. So the idea behind enzymes then is they're going to be very specific for what they combine to. And then the products they release can go on and do something else in our metabolism. Sometimes they'll go to another enzyme. Sometimes that'll be the finished product. It just depends on what the enzyme's goal is and what the body needs or the cell needs. Now, the process of trying to grab an enzyme, trying to grab a substrate, change it with an enzyme and release it sounds kind of clunky. But in reality, these can occur between 1 to 10,000 times a second. I mean, it's like really fast. So our enzymes are present and they're ready to grab things and move. So our cells are very, very efficient with the use of their enzymes. Now again, the enzyme is not destroyed, it's recycled, just like our key is recycled. So our body doesn't have to spend a lot of time recreating things. Once we have an enzyme, it's usually there for a while, and we can use it whenever we need it. Same with bacterial cells. So it's very efficient. The only time we have to worry about our enzymes is when we have something occur in our bodies that is changing things. Like maybe we have an instance where our blood becomes more acidic. That can affect our enzyme. Or we have an instance where we become overheated, like from a fever. That can affect our enzymes. So that's when we really have to worry about enzyme production. Otherwise, they're just there and ready to go. Now, in biology, rarely do things make sense. But thankfully, with enzymes, whoever named them did a great job because the enzymes actually make that. What's that? Oh, sorry. Let me get my chat down. Thank you for letting me know. Actually, I'll just have to close it. All right, I'll keep an eye on it though. Okay, so enzymes then are classified into 6 categories, and their names actually tell you what they're doing. You can always tell an enzyme because it ends in the last 3 letters of ASE. So you can always tell an enzyme because it ends in ASE. So we have hydrolases. Well, hydro sounds like it's involving hydrogen and the hydroxide, water. And it is. So hydrolases are going to be either bringing water in or splitting water. We have lysases. Lysing means to split or break apart. So these are enzymes that are going to split and break apart molecules. The difference between a hydrolase and a lysase is that they're going to use something besides water, besides H2O. Isomerases. Isomerases means that we're going to structurally rearrange the molecule. Okay? Now, isomerase means that we're not removing anything. It's staying the same. We're just rearranging it. Okay? Ligases, when you hear ligase, think of joining together. So we're making like a lignin or putting something together, making a long chain. Oxidoreductases, we're either oxidizing or reducing. And transferases means that we're moving things around. We're transferring functional groups. Isn't that a nice thing about enzymes? They all make sense. It's like one of the rare things in biology that you can tell what's going on from the name, right? Otherwise, it's very confusing at times. Now, the enzymes that are present are going to help move things along. And the process that they help move things along is because they do a couple of things with our reactions. First of all, when we learned about enzymes, we developed what's called the collision theory. And the idea behind this is that if I put a whole bunch of chemicals into a test tube or into a broth, they would eventually bump into each other. You know, it's like if you just eventually, just out of sheer luck, things would bump into each other and reactions would occur. Enzymes, though, help make sure that those bump into each other actually occur. The enzyme speeds up the process. It takes away that chance that they might interact and actually makes them interact. Think of it as kind of like, what do you call it, matchmakers, right? You might run into the love of your life, but a matchmaker will help you find somebody. Okay, that's kind of what an enzyme does. It's like a little matchmaker out there. All right, so it's trying to bring molecules together and change them. Now, the thing about this then is we have a certain amount of energy that's required for a chemical reaction to occur. We call that our activation energy. That means I have to put a little energy in to make something happen. Now, the nice thing about our enzymes, we can see here, is that in peach, or that pink color in the background, that's showing how much activation energy has to be present to be able to have enzymes to be able to have substrates bind each other. But check it out. In purple, it shows when the enzymes are present, how that activation energy drops. It's kind of like if I'm rolling a boulder up a hill, the enzyme makes it so that my hill is like really, really small or almost flat. It takes away that activation energy. The reaction rate is how likely I am to have molecules come together and interact. As I said, an enzyme's like a matchmaker. So what it's going to do is it's going to increase that reaction rate. It's going to make it much more likely. So we're getting two things here. We're getting a decrease in activation and increase in reaction. Okay, decrease in activation, lowering energy, and an increase in our reaction rate. Things are going to happen faster. So enzymes are going to help us with different processes in the cell because of that. Now, when we look at enzymes, a lot of enzymes, when they're made in the cell, are complete and ready to go as soon as they're put together. And these are great. They're all set and ready to go, and our cells can use them right away. Other enzymes, though, are going to need something else to make them functional. We call those apoenzymes. Apoenzymes mean that we have a protein that's a part of an enzyme, but it needs to have something else to make it active. So in other words, it's kind of like making a key, but I haven't done the form of the key yet. So you know how like when you go to get a new key, they have a form and then they cut the ridges in? An apoenzyme is like that key that you just pulled off the shelf. And then when they cut in those ridges, that's when it's going to be the enzyme. Now, what is needed to fill those ridges in enzymes are going to be things like a cofactor or something like a mineral or a vitamin. Those are like things of cofactors. So this is why, like in our diets, we have to make sure that we're bringing in vitamins and minerals, is because that helps shape our enzyme correctly, just like how we shape the key correctly. And so that makes the enzyme usable. So if I have an apoenzyme and I bring in the correct cofactor, they work together and they create what's called a holoenzyme, which means now it's a whole enzyme ready to go. Okay, so it's just like if I get a key, like kind of model, I cut it to shape of my key, and now it's ready to go. So my finished house key is my holoenzyme. All right. So here's a picture then showing that. I have my enzyme, which is my apoenzyme. That's the protein piece. I need to have a cofactor. That cofactor could be a vitamin. It could be a mineral. It could be something like that our body else has to make. It's going to hook together with the enzyme, and now it's ready to interact with the substrate. Now it's ready to go. We call that the holoenzyme. So with our enzymes then, they're going to be really important to help processes happen. We have a couple of cofactors that are also going to be really important. Our cofactors are going to help with our redox reactions. So remember, redox means that we're losing electrons and gaining electrons, right? So redox reactions means both of those are happening at the same time. So the idea behind this with these cofactors is that something is going to lose an electron, and these cofactors are going to grab the electrons that were lost and move them to another part of the cell process. You can think of these a lot of times as being almost like an Uber driver, how they shuttle us around. That's what these guys are going to be doing. These are going to be our main cofactors. So the main ones that we have are NAD and FAD. And NAD and FAD are going to be important in our metabolism because their job is to grab electrons and to move them to somewhere else in the cell to be processed. So again, kind of like a little Uber driver just shuttling electrons around. So what's going to happen then with our NAD and FAD? My NAD is going to be able to be oxidized or reduced. And we can tell what form it is based on how it looks. When I have NAD present, NAD by itself, that's its oxidized form. So that means it doesn't have any extra electrons, okay? That NAD can now come into contact with an electron. And what we see when we shuttle electrons around is a lot of times the electrons that are removed are ones that are usually associated with hydrogen molecules. Now remember hydrogens. How many electrons does a hydrogen have? One, excellent. And so the idea behind the hydrogen is it either wants to be in a bond to fill its shell, or it's also willing to kick that electron off. We use hydrogens as electron sources in metabolism because they're easy to pull electrons off of. Number one, they're smaller molecules or elements, so we can pull things from them. And number two, they tend to easily give those electrons away. They don't fight it. So when we talk about electron shuttling, what we see is that a lot of times when we're moving electrons around, you'll see that they'll be represented by moving with a hydrogen. And you might be going, well, where's the hydrogen coming from? It's because it's the electron donor. Sometimes what happens is the hydrogen will give its electron away. It becomes a hydrogen ion, so it becomes more positive, and then it becomes attracted to the molecule that took its electron. Because remember, whatever took its electron becomes more negative. So that's what you see here. NAD in its oxidized form, it can pick up a hydrogen as well as a pair of electrons. And that makes it become NADH. NADH means it's reduced, meaning that it has gained electrons. So you can tell which way it can go based on that. So if I see NADH, that means it's full. My Uber driver is full. Okay, if I see NAD, that means the Uber driver can pick up passengers. Okay? So you'll know we have the same thing with FAD. And FAD is able also to pick up electrons and a hydrogen, and it's going to become FADH. All right, so the same idea, just whether or not it has room to pick up our hydrogens and our electrons. Now, the reason this is important is when we talk about metabolism, we're going to see NADs and FADs. We're going to see them swooping around and becoming NADHs and FADHs. It's just showing you what's happening and where electrons are going. Okay, so we're kind of like learning the players of the game here. So those NADs then, they're able to accept electrons. NADH means they're full. FADP is another one. We see a lot, NADP is another one. We see this a lot of times with plants. They tend to use more NADPs than NAD. It's just another cofactor or coenzyme. FAD, as we said, can pick up electrons, so it can go from FAD to FADH. And then another electron carrier is one that's referred to as coenzyme A. Now, you might look at that and go, coenzyme A, it says it's an enzyme, so that means it's an enzyme. But look at that name. Does it end in ASE? No. And what's the rule with our enzymes? They all end in ASE. So this is one that we at first thought was an enzyme, and then we found out later, no, it's not really an enzyme, it's more of a cofactor. It's needed to help make things happen, but it's not a true standalone enzyme. So it's one of those ones that's a fake one, all right? So don't believe that this is an enzyme. If you see that ever pop up, hey, is coenzyme an enzyme? No, don't believe it, because it doesn't end in ASE. Okay, so the reason that we're looking at enzymes then is they make things happen, and this brings us back to that idea of why protein folding is so important. We talked about the difference between baseball gloves and oven mitts with protein folding. The same is true with our enzymes here. They have to be folded correctly to make processes happen. Just like our house key has to be made correctly. Have you ever had a key that you cut and then they didn't cut it just right and it didn't work? Am I the only one that had that happen? Okay, good. That makes me feel better. I was like, am I the only unlucky person? Yeah, you try to put it in the lock and it just doesn't work. They have to redo it. So that's the same that's true with our enzymes. If we fold them wrong, it's like having the wrong key. It's not going to work. So a lot of our genetic illnesses that are out there are ones that involve enzymes that are not folded properly. And depending on what part of the process they're affecting, we're going to see that effect more clearly, or it might be more hidden depending on what kind of genetic disease they have, what kind of things are going on with their enzymes. Now, things affecting our enzymes, as I mentioned before, temperature. So what we find is that if we elevate the body temperature a little bit, that can actually speed an enzyme up. Like when we're working out and our temperature is increased a little, our body will actually have the enzymes move a little faster, just like when we heat a molecule, they'll move a little faster. And that allows our reactions to occur quicker because when we're working out, we need energy faster. But if we push it too far, like with a high fever, we're talking about like 106, 108, we're getting too high, it can cause our enzymes to start basically melting or falling apart. So temperature is going to be very important. The same is true in the opposite way. If we run into a situation where we become too cold, hypothermic, we can run into an issue where our enzymes no longer function correctly. They have a harder time transporting blood. They have a harder time staying warm. And that can result in tissue damage. You've heard of things like frostbite. That's because we had enzymes that were damaged, especially in the extremities. Okay? So we actually use this nowadays to our advantage. One thing that we've learned, and if you've ever worked with an ambulance driver or if you're ever in an ambulance, depending on what kind of call they're doing, they have ice blankets. Have you guys heard of the ice blankets? A few of you are nodding. So if you pick up a patient, depending on what kind of trauma they have, a lot of times what they'll do is they will actually lower the body temperature of that patient. It slows down the enzymes and it actually slows down the internal trauma. And so you have a little longer to get them to a hospital because it's stopping the reactions that are occurring in the body, and it can actually delay shock and it can delay internal issues. So it's really fascinating. So a friend of mine was in a really bad accident. They had to use the jaws of life on her. And she said she was so happy to get out of that car. She was so relieved. And she's like, can I get in the ambulance? And I'm like, okay, things are going to get better. And then they put an ice blanket on her. And she's like, put me back in the car. At least I was warm in the car. Like, this is ridiculous. But they were trying to lower her body temperature to prevent shock. And it helped her. She survived, thankfully. So the idea then that temperature is important is very true. Another thing is pH. We don't want to have things too acidic or too basic. Now, keep in mind that's also going to depend where we are in the cell or in the body. So things in our stomach, for example, they're going to be used to a high acidity or to an acid type of pH. If I move those enzymes from my stomach to another area, they won't function. So the enzymes are made to function in their specific pHs. And then substrate concentration, the things that the enzyme interacts with. If I have a lot present, my enzymes work better. If I have very few present, it slows the enzymes down. The chance of them bumping into each other becomes less. And then inhibitors. As I said, enzymes are always around. They never get destroyed. So an inhibitor helps turn an enzyme off. It basically says, okay, we don't have to worry about making any more of this, whatever we were trying to make. So I need to shut the enzyme off for a while until I need more, and then I can turn it back on. Okay, so it's a way to shut things down and make sure we're not wasting energy. Here's a picture of an enzyme that has been denatured. So we call this term denaturing whenever an enzyme has been damaged in some way. And again, this is usually related to heat or to cold or to pH, being acidic or basic. It will cause my glove that I've made to basically melt and fall apart. So now my enzyme doesn't shape correctly, it doesn't react correctly. So we'd say that's denatured. We purposely denature food or enzymes all the time when we cook food. It's like when we scramble an egg. The reason the egg changes its shape is we're denaturing the enzymes that are present. That's why it becomes fluffy and solid as opposed to that kind of liquid kind of appearance when we first start. So that's a change of proteins or enzymes. Now, as I said, one of the things we want to really be able to do is to regulate our enzymes. And we have to be able to turn them on and off. And there's two different ways that we can do this. We can use something called allosteric inhibition. This is also sometimes referred to as non-competitive inhibition. And so this is where we're going to have something where we can turn the enzyme off because we change the way that it appears. We change its shape. This is like, changing your key, putting an extra notch on your key temporarily, so it turns the enzyme off. Now, this is going to rely on negative feedback loops. And in your other science classes, I'm sure you've heard of negative feedback loops. It's basically where I have a process occurring, and then I get a negative feedback loop that stops the process and pushes it the other way. A positive feedback loop is where I just keep going and going and going with the process. So negative feedback loops are very important. They stop a process. So what's going to happen here is I'm going to end up with an enzyme that's going to be turned off. A lot of times the things that turn the enzymes off can be something like the product that they manufacture. So let's say I have an enzyme reacting with substrate A, and it's releasing it as product B. Okay, so I take something and I change it to a final product. As I keep doing this, as I move from A to B, A to B, A to B, I'm going to end up with a bunch of Bs. That B product, as it builds up, can over time go and attach to the enzyme and turn it off. And now the cell can use the Bs, which means that my B numbers will decrease as the cell uses them. And as it gets down to the last B, it'll pull that last B off of the enzyme, and now the enzyme's ready to go again. See how that works? So I can add things and I can pull them off. So with that then, what will happen is if I have a substrate that is interacting with my enzyme, like I said, the letter A, for example, interacting with my enzyme, as I build and release more of these letter Bs, that letter B will eventually shut off the enzyme. The letter B in this case is represented with that little yellow color. Now, this is an example of allosteric inhibition. It's also called non-competitive inhibition. And the reason for that is because the B, or the inhibitor that's turning it off, is binding in a different location than where the substrate is binding. See how that works? It's binding, like, for example, on the bottom of the enzyme. So it's not competing for the site. Okay, so that's different. So allosteric inhibition, non-competitive, they're not binding in the same place. They're not competing for the same place on the enzyme. But what happens is when it does bind to the other place on the enzyme, it's going to turn the enzyme off. All right. Competitive inhibition is where I have an enzyme that'll be turned off because I will have a substrate come and sit in the area that it's been interacting with the other substrate and will turn the enzyme off by changing its shape. This is considered to be competitive because they're binding in the same spot. So same idea, let's say that my substrate in red is my letter A, so I'm producing A's and I'm releasing them as B's. And the yellow spot, notice this time instead of binding at the bottom of the enzyme, it's binding in the same place where the A was. So that's considered to be competitive because they're fighting for the same spot. Once that B binds, it sits on the enzyme and it turns the enzyme off. Now, the idea behind this, and the reason we're able to regulate our enzymes, we don't want them doing a process when we don't need to. So in other words, I don't want my body making a whole bunch of ATP because I don't always need a whole bunch of ATP, right? It's kind of a waste. So I need to be able to regulate, to turn my enzymes on or off depending on my needs. Now, we use these competitive inhibitions a lot of times with things that will control how enzymes work. So for example, penicillin. We said before that it affects cell walls. Well, the reason it affects cell walls is it competes and binds in the place of the substrate that the bacterium needs to make a cell wall. So my penicillin goes and sits on their enzyme, and then that enzyme doesn't work anymore. And the bacteria cannot build their wall effectively. I also can use this with things like ibuprofen. When we take an ibuprofen, it's going to stop, for example, inflammation responses. It's going to stop pain responses. It goes and attaches to our enzymes that are involved in those processes and turns them off. It lasts for a few hours, it wears off, we take another ibuprofen. So the ibuprofen is fitted to sit on those enzymes that are involved in that process. Cancer drugs. So things that we're going to take that are going to be used to try to stop cancer growth. They're going to stop the enzymes responsible for cell division, like mitosis. That's why chemotherapy is so hard. When you go and you get dosed with chemotherapy, what's happening is we're shutting down your cell division. This affects the cancer area, but it also affects things like our white blood cells, which is why our immune level can drop. That's why we have to be careful when we're doing any kind of cancer treatment, because our immunity will lower. So all of these things are a factor. Another one is the drug Lipitor. You might have heard of that. It's a drug that's used to lower cholesterol. So the way that it works is when you take Lipitor, it binds with the enzymes that are in charge of making cholesterol, and it turns those enzymes off. So your body does not produce cholesterol. That lowers your cholesterol number. The problem that we found with Lipitor is if you know a loved one who's taking it, if they start having leg cramps, they need to stop taking Lipitor right away. The reason for that is we found that in some people, the Lipitor can bind the cholesterol enzyme, but in a few people, it can also bind an enzyme responsible for muscle relaxation. And so what it does is it makes the muscles stay tense, which is why they start to have leg cramps. Their muscles are constantly contracting. And so it's one of those things where if somebody's having that effect, they just need to change what kind of drug they're using to lower cholesterol. They need to change something with a different shape. And that's what a lot of our drugs out there are. They're just different shapes. That's why we have things like Bayer and ibuprofen and Advil. They're all doing the same thing. They're just different shapes slightly. And so for some people, they work better than others. It's all about our enzymes. Okay, let's check our understanding. Enzymes are going to increase the speed, so make things happen, by lowering activation energy. Do they lower the hill? Do they lower the activation energy? Is that a true or false statement?
True.
True. Yeah, I see lots of nods. That's a true statement. So we know that A is correct. That means that B is not correct, right? Just by answering A. C, do they increase the frequency of collisions?
Yes.
Yes. So C is correct. That means that D cannot be correct. Okay, so the correct answers are going to be A and C. So they're going to lower the hill, the activation energy, and increase the rate of reaction. What happens to an enzyme when it is above or below its optimal temperature? Do we say it's been vanquished? Do we say it's been destroyed? It's been denatured, or it's been discarded?
Denatured.
Denatured. Excellent. So we say that it's been denatured. And what is allosteric inhibition? Is it binding where the substrate binds? Is it binding where the product binds? Is it binding in a different location for the substrate? Or is it binding in a different location for the product?
Different location.
C, excellent. So allosteric, remember, is non-competitive. It's binding in a different location than where the substrate is going to bind. Excellent. Okay, so when we start looking at our next step of slides, we're going to be going through the typical metabolism that we see in most cells. And so the idea behind this is we're going to follow some metabolic pathways. Now, the big picture in the bottom line of the metabolic pathway is that when we bring in food molecules, we are going to start by oxidizing that food molecule. So what does that mean is happening to the food molecule? It's being oxidized. Excellent. It's losing electrons. So when we eat food, one of the first things we're going to do is start pulling electrons off of it. Okay, so we start our process with oxidizing. These electrons then are going to get moved through our metabolic process. And their movement and later on the flow that they're going to create are actually going to be what generates or drives our ATP production. So we need to have constant inputs of food to be able to be allowed to produce constant ATP. If we run out of food, we start running out of problem where we don't have enough ATP. Just like if you've ever gone too long without eating, how do you feel? Sluggish, tired, right? If you go too long, you just feel kind of like exhausted. You feel good. You feel angry. Yeah, hangry. A lot of people get hangry, right? Yeah, exactly. Like it makes you grumpy, right? Okay, so the ATP then is going to be our ultimate goal that we're going to be looking for when we do any type of metabolism. And remember, ATP is going to be a process where we need to be able to make a molecule that has 3 phosphates on it to make ATP. The way we do that is we're going to take an ADP and put a phosphate back on it. Remember, we take ATP, we use it, it becomes ADP. The way we go back is we put a phosphate back on. That process, though, of putting a phosphate back on is going to take a lot of energy. And that's what's going to be involved in metabolism. Our metabolism is going to drive or provide energy to shove phosphates back together. That's going to be what we need to be able to stay alive. Now, in our cells and in most bacterial cells, the main process of creation of ATP is going to occur in the final process of metabolism, which is called the electron transport chain. This is where we get the most phosphates put back on our ADP to create ATP. So this is going to be our main process. And what we're going to see is that all the metabolic processes before the electron transport chain are kind of being done to set up the chain process. So this is kind of our big one. So ATP then, remember, it's our energy source. During catabolism, energy is released, and it's going to do that by pulling phosphates off. And then at the end, we have to redo or recreate ATP, which means we have to put a phosphate back on. Now, our general metabolism then, for most cells, our cells, bacteria included, is going to be something very simple like what we see at the bottom. At the bottom, it's saying that we're going to take a glucose molecule, C6H12O6. That's glucose. We take a glucose molecule. We're going to add some oxygen to it. Okay, we're going to need oxygen for our respiration. Aerobic bacteria are going to need oxygen. The only difference is a few little anaerobes who don't use oxygen. We'll talk about those. We're going to convert our glucose with that oxygen into CO2, some water, and some ATP or energy. That's going to be our net process. So we're going to go and add phosphates back to ADP. We're going to walk away with energy, ATP, and we're going to walk away with a little water that's going to be produced. So we're going to go through the processes, the main process, step by step, so that we understand each step of the process. And we'll talk about a few variations with our bacteria. So the three main stages in carbohydrate metabolism, which is where we're breaking carbs down to create energy, is we're going to have glycolysis, we're going to move to the Krebs cycle, and then we're going to have the electron transport chain. Now, the glycolysis, we're going to see that we actually have three different pathways we can follow that our glucose molecule can go through. Two of these are the same for us, and one of these is unique to bacteria. Okay. Once we're done with glycolysis, we end up with a product that has to go through a conversion. We call that the intermediate step to get the product ready to go into the Krebs cycle. Then we go through the Krebs cycle, and then we go right into the electron transport chain. And remember that ETC is where we're going to get the most ATP. Oh my gosh, all these acronyms, huh? ETC, ATP, OMG. It's like, oh, trying to keep track of it, right? But don't panic. It's just acronyms. Don't get stressed out. So what we're going to do with glycolysis, we're going to have a partial oxidation. Okay, let's remember what that meant, losing electrons. We're going to lose some electrons, and we're going to, at the end of glycolysis, end up with a molecule called pyruvic acid. You also might hear this referred to as pyruvate. They're used interchangeably. Okay, we're going to make a little tiny bit of ATP, like a small amount of ATP. And because of the fact that my glucose is losing electrons, that means the electrons have to go somewhere. The electrons are going to get gained by our Uber driver, NAD. And when it gains the electrons, it's going to go from NAD to NADH. Everybody with me on that? That's our simple overview. Okay. So the glycolysis pathway, or the glucose pathways that can occur, we can have the one that we are going to see in most eukaryotes, and that is referred to as the EMP pathway. And that's for Embden-Meyer-Parnas pathway. Those are the three scientists who figured this out. So we refer to it as EMP for short. This is the one that we use. It's the ones that most bacteria that are out there use as well. We're going to have the pentose phosphate pathway. That's going to be one that we use as well. And bacteria use to create nucleic acids. So that's where we're going to basically set the precursors for DNA and RNA. And then what we're going to find is we have a third pathway, the Ettner-Dwardoff pathway, that is unique to only a few microbes, a few bacteria that are out there. What we find is that a lot of times these microbes that are doing this pathway, it's like an older or an ancient pathway. So we think it's something that probably early eukaryotes used too, but they later evolved to the EMP pathway. And they changed. It's more efficient. But we have a few holdouts. You know how there's always that one person who will not embrace technology? There's a few bacteria that are like, we're not doing it yet. It's only been here for a couple million years. I need at least another million. So they're staying with this old pathway. And that's going to be the Ettner-Dwardoff. So let's look at glycolysis, the EMP pathway. So again, this is going to be the main one. This is the one that we use. It's the one that most prokaryotes use. So what's going to happen is that this process can happen with or without oxygen. So it's going to occur under an anaerobic condition, meaning oxygen does not need to be present to have this process occur. If we're a prokaryote or a eukaryote, it's going to occur in our cytoplasm. So in the same location for both, in the cytoplasm. What we're going to do is we're going to take a glucose molecule. From that glucose molecule, we're going to break it, rearrange it, and when we're done, we're going to have some electrons that have been moved to NAD to create NADH. And we're going to end up with that glucose being converted to form a pyruvic acid or pyruvate molecule. So we change the shape of the glucose forever, all right? And then from there, it can go on to an intermediate step in the Krebs. So when we look at this process, what we're going to see is that it's a process that doesn't produce a lot of ATP. It actually requires ATP to get started. So one of the things we look at when we're looking at metabolic processes is what their net output is. So in the case of glycolysis, we have to put 2 ATP in. We get 4 out. So if it's 4 minus 2, what's my net? 2. So it's a lot of work for only 2 takeaways, 2 ATP takeaways. So it's a lot of work. So I have some energy investment. I sacrifice some ATPs. I get some breaking and rearranging of my glucose. And then when I pop the glucose out, it is now forever going to be pyruvic acid, at least for the end of glycolysis, and I get some energy to create ATP. Here's the process. Isn't that easy? Just look at that. That's so easy, right? Okay, so let's look at this for a minute and be like, what the heck is going on here? Okay, the black circles are representing carbons. And so this is showing your carbon chain of your glucose. So the first step, step 1, is where we're going to have glucose being oxidized. Remember, that means losing electrons. And we're going to be converting it to our pyruvic acid. Then we're going to have those electrons go to our carriers, our Uber drivers, so our NADs. And then we're going to produce some ATP. Now, this process then, we start with a glucose molecule. We're going to sacrifice some ATP. You can see at the top, there's 2 ATPs that get sacrificed. The reason they get sacrificed is they end up putting a phosphate on each end of the glucose molecule. This is the commitment that the cell has to go through. And the reason we put a phosphate at the end of the glucose molecule is to prevent it from leaving the cell. Glucose can come in through diffusion. It's not charged. But we need to stay in the cell. So one thing we know with plasma membranes is if it has a charge on it, it can't leave, right? So if you want it, instead of put a ring on it, you got to put a charge on it. And that's what our cells do. They put a phosphate on it, all right? So we can change Beyonce's song to if you want it, you put a phosphate on it. Okay, so we keep it in the cell now. Now what we're going to do is we're going to take that glucose complex with the phosphates on each end, and we're going to break them apart. Now remember, glucose, we said, has 6 carbons. So when I break them apart, I get 2 3-carbon chains. Now I go through a bunch of rearrangement, and then what I'm going to do is I'm going to have those phosphates that I put on there pop back off. That's going to create some energy, which is going to create some ATP. So I get 2 ATPs for the phosphates I put on there. I have some substrate level phosphates. What that means is they're phosphates that are just around the cell that come in and attach to help keep it charged. They pop off. So when those pop off, I get 2 more ATPs. So now I have 4. But I sacrifice those 2 to start. So the ones I walk away with, my net, are only 2. Okay, so we'll go through this process, don't worry. So the plus side of glycolysis is it's a very fast reaction. We're cutting glucose in half. We're creating some energy. We create 2 ATP net. No oxygen is required. But the minus side, or the bad side of it, is that we don't get a lot of ATP. Now let's go through the process of it and just kind of visualize it. Okay, glucose comes in. Glucose is usually as a ring. So I'll go back to the previous slide so you can see our process here. Glucose comes in as a ring. We open it up. We phosphate one side. We phosphate the other. We split. Phosphate this side, phosphate the other. And now I have 4 phosphates in play. I do some rearrangement, pop phosphates off, and I get some ATP. When I'm done, I have 2 3-carbon chains. Those are no longer glucose. These are now referred to as pyruvic acid. Okay? So you can think of this as kind of like a cool club dance. We go glucose, we open, we phosphate, we break, we move apart and rearrange, we get some ATP, and we end up with 2 pyruvic acids. Don't you guys love that? It's like this cool club dance, right? I mean, you can kind of picture it in your head. All right, the reason I show you that is because sometimes when we get our exam, we blank, and then I'll see students doing that. And it's like, that's good. You know, you're visualizing, and that's great. So visualize the process in your head. Now, that's one way that we can process glucose. But I told you there's 2 other processes we can use. One of them is the pentose phosphate pathway. What's going to happen here is glucose is going to come in, so it's a little 6-carbon. But what we're going to do is when we open it, we're going to pull one of the carbons off. And we end up with a 5-carbon. So once we have that happen, that 5-carbon now can become the basis of the 5 carbons that we see with nucleic acids. This is a process that we can use when the body needs to make more ATP or more RNA or more DNA. We just need to create a 5-carbon sugar. So we get a 6-carbon sugar in, glucose, we break a carbon off. So it's going to go down a different pathway. This one isn't really going to generate energy. It's instead going to generate nucleic acids. So we're just rearranging to get some nucleic acids out. And we'll talk about the ED pathway in a minute. Okay, so the pentose phosphate pathway, the PPP, is what we're going to use to create pentose sugars. Pentose meaning 5. Okay, so we have the pentagon, it's in the shape of 5, right? Pentose meaning 5. So what we're going to do here is we're not going to create any energy in this process. Instead, what we're going to be doing is we're just rearranging carbons and removing 1 carbon to create a 5-carbon base. So what's going to happen is we're going to have glucose come in. We refer to that glucose as G6P. What's going to happen then is we are going to rearrange. So we're going to move some electrons off of that glucose. We're going to get oxidized. And then what we're going to do is we're going to pull a carbon off to create a 5-carbon ring. And that 5-carbon ring is going to be the basis of our DNA, RNA, or ATP. So if we look at this process then, glucose comes in. We phosphate it to make sure it doesn't leave the cell. But then what we're going to do is instead of continuing through that process towards pyruvic acid, we're going to move to a side pathway. And a side pathway is going to remove a carbon. And when we remove carbons, they usually get removed as a CO2. So we break that carbon off and attach it to an oxygen. And then we're going to move over into a different pathway, which is going to create a 5-carbon sugar of DNA, RNA, or ATP. Yes?
I thought the pentose phosphate pathway was more of like a RNA, so nucleic acid rearrangement, right?
It is. So that's what this is showing. So it's showing we're making a 5-carbon. And remember, 5-carbon is the base of DNA, it's the base of RNA, and it's also the base of ATP.
So it doesn't need energy, but it still works with PTP?
Correct. So what we're doing is, remember, we have to sacrifice an ATP to charge the molecule so it doesn't leak. But then we rearrange it. But remember, phosphates are in DNA, phosphates are in RNA, and phosphates are in ATP. That phosphate stays with it.
Okay.
It doesn't pop off to create energy. We keep putting energy in, but we're not popping the phosphate off to create an energy. Great question. I love that. That's a great question. Does that make sense then, what's happening there? So it's just rearrangements. Okay, and then the third pathway that our glucose can take is referred to the Eppner-Dor pathway or the ED pathway. This is going to be, again, what we see in a few bacteria that are old school. We see these in bacteria like Pseudomonas and in Enterococcus. Pseudomonas is one that can cause ear infections in people. It's also associated with water. And then Enterococcus faecalis is one that can cause urinary tract infections in people, and it is also associated with GI upset. So this process is unique to prokaryotes. We cannot do this process. What's going to happen is we're going to have glucose come in. We're going to phosphorylate the glucose. But then, instead of processing both sides of the 3-carbon molecules that we create, we split the carbon in half and create 2 3s. Instead of processing both sides equally, the bacteria process one side, and then the other one goes through unchanged. So it's kind of funny. It's like they do kind of a half process. What this means then is that they are not going to generate the same amount of energy as what we see with our normal glycolysis, or with our pathway. We create 2 ATP net, they create 1, because they only do one side. We create 2 NADHs, they create 1. So it's basically like taking our process and cutting it in half. That's why we think it's older. We think that probably in the beginning they didn't have enough enzymes to do both the same, and so they just kind of limped along with this process. It still works for them because they're so small, but it wouldn't work for us. Our cells need more energy. So we think that they have, and we know they have different enzymes. They don't have the same enzymes needed to process the EMP pathway, which is what we do. So here is showing our normal glycolysis. So our EMP, you can see where we separate our glucose. We then go through the 2,3 carbon chains, go through the same processes on both sides. That's our pathway, that's EMP. The Etner-Dwardoff pathway, we get separation, but then we have one of the molecules that just goes straight to pyruvate. The other one gets processed and it releases the phosphate that was added to it, but we only get 1 ATP out then. So it's just like doing the process halfway. It's like it gets tired, basically. It's like, who has time to make, 2 molecules? I'll just do 1. And because of that, it walks away with 1 ATP. When we walk away, we have 2 ATPs. Okay, so it's just basically doing like half the process. Okay, we good? Okay, so remember, this first part of the process, glycolysis, it's going to occur in the cytoplasm for eukaryotes and prokaryotes, and it's going to occur without oxygen. I don't need to have oxygen present to be able to do this process. For organisms that are strict aerobes, organisms like us, they have to have oxygen to live. We can't go to the next step of metabolism unless oxygen is present. So if we don't have oxygen present, we get stuck in glycolysis. The same is true for certain types of bacteria. They don't have oxygen present, they just keep doing glycolysis over and over and over again. We call that process fermentation. So fermentation is where we keep repeating glycolysis. The idea behind it then is that we're not creating a lot of energy. We're just creating 2 ATP each time. What we want is we want to have oxygen present so that we can create more energy. If oxygen is present, we'll go on to the next steps. Okay, so the next steps are going to be where we go through the intermediate step and we go through the Krebs and the electron transport chain. Now, for strict anaerobes, like our Clostridium, we mentioned that oxygen is toxic. So what's going to happen for them is they can't have oxygen present to move on. Instead, they're going to have another molecule present to move on. So for them, it could be something like a nitrogen or a sulfur, something along those lines. And each one will have a specific molecule that they're built around. Remember when we talked last time, I talked about how if we have a sheet that we're holding at the 4 corners and we put a bowling ball on it, that's going to pull things to happen. That's why we have to have oxygen present. Oxygen is our big bowling ball sitting on the sheet, which is going to pull molecules through glycolysis, through the intermediate step, through Krebs, and down into the E2C. As they fall towards that oxygen, they're going to move through these processes and create ATP. If I don't have oxygen present, I don't have a pull. If I take that bowling ball off the sheet, I have nowhere to go. That's why I have to have oxygen to keep pulling. For my strict anaerobes, that bowling ball, instead of having oxygen written on it, will have sulfate or sulfurs or nitrogens. Okay, it's whatever's pulling things through. All right? So for us, the next step of our process then, for us aerobes, is that the pyruvic acid that we created, remember that's 3 carbons, is going to be oxidized. Remember that means it's going to lose electrons. And what's going to happen is we're going to remove a carbon from the pyruvic acid. And remember, when we remove carbons, it usually gets released as CO2. That's why we breathe out CO2. These are extra carbons we don't need. And we're going to rearrange what our ending molecule is. We're going to put it into the Krebs cycle, rearrange it. And then when we're done, we're going to be ready to go on to the electron transport chain. So exciting. Our final acceptor at the end of the chain for us is going to be oxygen. That's the one that, again, is the bowling ball making this whole process flow. Okay, so we're trying to get electron flow. So with this then, we already did our glycolysis. We ended up with 2 pyruvics or 2 pyruvate acids. We now are going to need to go into the Krebs cycle, but we're stuck. And the reason we're stuck, the Krebs cycle has a bouncer in place that only lets a 2-carbon molecule enter inside. With our pyruvic acid, it's a 3-carbon. So what that means is we have to pull a carbon off to create a 2-carbon to enter into Krebs. So that's the intermediate step. It's going to pull a carbon off, release it as CO2, and now I have a 2-carbon chain that can go into Krebs. All right? When I go into Krebs as my 2-carbon chain, I can go through the rest of my process, and then I can start my electron transport. So what's going to happen in that preparatory, or it's sometimes referred to as the intermediate step, is I'm preparing to go into Krebs. I can't go in there with a 3-carbon chain. The bouncer says no. I have to make it a 2-carbon chain. So I pop a carbon off, it leaves as CO2. When I break that carbon off, I'm going to release some electrons. So when I do that, those electrons are going to get picked up by NAD to make it become NADH. Okay, so I'm breaking a few bonds, I'm releasing electrons. I now am going to create a 2-carbon molecule that I'm going to refer to as acetyl-CoA. Acetyl-CoA means I've converted the pyruvic acid from a 3-carbon to a 2-carbon, and when it's a 2-carbon, it becomes acetyl-CoA. Okay? The acetyl-CoA now meets the regulations of the Krebs bouncer. It's a 2-carbon molecule, so it can now go into Krebs. And so now I'm ready to go into the Krebs cycle. So this is all occurring in my little intermediate or preparatory step. Okay, once I'm in the Krebs cycle, what's going to happen in the Krebs cycle is I'm going to do a lot of rearrangement with my molecule. Basically, I'm going to add carbons back to it. I'm going to break carbons off again. I'm going to rearrange the carbons. I'm going to move them around. And the whole reason I do Krebs with these additions and breaking and rearrangement is every time I break a bond or rearrange a bond, I'm able to shuffle hydrogens and get electrons out of it. That's the whole point. It's kind of like building Lego bricks and then breaking them down and building them back up again. So my cycle is constantly adding bricks, breaking them off, adding bricks. When I break a brick off, I get some electrons. That's what I'm trying to do in the Krebs. I'm basically just rearranging things to get ready for the ETC. Now, the Krebs cycle is referred to as the Krebs cycle because it was found by Hans Krebs, who was the first one who figured it out. It's also sometimes referred to as the citric acid cycle. And that's because the first molecule that we're going to make in there is going to be citric acid. When we go in there with the acetyl-CoA, it's going to have carbons add to it to create citric acid. It's also sometimes known as the TCA, the tricarboxylic acid cycle. And what that means is that we have one step where we add carbons and create a tricarboxylic acid. It has a lot of different names. It's the same process. The reason I tell you that is if you go on and you take a test, maybe somewhere along your life, and they ask you about citric acid, or they ask you about Krebs, or they ask you about TCA, you know that that's all the same process. It just has different synonyms that are associated with it. When we're done with Krebs, as we start to work our way through, for each acetyl-CoA that enters the process, we're going to walk away with 1 ATP net, 3 NADHs, and 1 FADH. We're going to walk away with those net. Now, remember though, how many acetyl-CoAs do we have that are going to be entering the process? How many pyruvic acids do we end up with from glycolysis? 2. So remember, these numbers, the 1, the 3, and the 1, if I'm going through the entire process, we're going to times it by 2 because I have 2 molecules. All right? Because I have to go through this process twice. So let's kind of draw this out for a minute to make sure we're doing okay. I have glucose. I'll start with glucose. Glucose is going to come in. Now, glucose is a 6-carbon sugar. Glucose is going to come in. What's the first thing we're going to do with glucose? We're going to open it up.
Well,
Okay, that's okay. And then what are we going to attach to the end?
phosphate.
Phosphate. So I lose a phosphate. That's one ATP. What do I attach to the other end? Another phosphate, right? Okay, so now I've charged the two ends. What happens next with my molecule? Breaks in half. So now I go from a 6 carbon to 2 3 carbons. All right? And then I have some ATP near the environment that are going to come through and they're going to add phosphates. So now I have phosphates that are on each end. Okay, so I have 2 molecules, each end has a phosphate. Now I'm going to do rearranging and I'm going to pop those phosphates off. When the phosphates pop off, they generate a little bit of energy. Okay, so this phosphate flies off and it now creates ATP. This phosphate flies off, it creates ATP. How about these two? Same thing, ATP, ATP. So I get 4 ATP. But do I really walk away with 4 ATP? No. How many did I sacrifice to get started? 2. So I have to minus 2, and I get a net total of 2 ATP. That's for my glycolysis. Okay? So a net total of 2 ATP. I also now have these 3 carbon chains, right? And if I have a 3 carbon chain, it can no longer be called glucose. It has to change its name. What do we call it instead?
Pyruvate.
Pyruvate or pyruvic acid. You got it. So pyruvic acid or pyruvate. Okay, so that's going to be the same. All right. Now I told you that when we rearrange this molecule, we also are going to generate some electrons that get released. Why? Remember, anytime we're breaking a bond, we're breaking something off, we have the potential of having some hydrogens get released because we're releasing a bond. When those hydrogens get released, as I mentioned several slides ago, they have electrons that they're willing to donate, right? This is where we get also our 2 transporters that form our NADH. So we have 2 NADHs and 2 ATPs when we're done with glycolysis. Okay, I've got my 3 carbon pyruvic acid. It wants to go into the Krebs cycle only if oxygen is present, and can a 3 carbon fit in the Krebs cycle? No, remember the balancer at Krebs says, nope, I don't do 3 carbons, I only want a 2 carbon. So now I have to rearrange my 3 carbon. Okay, so the easiest way I'm going to do this, I'm going to kick a carbon off. So now I'm going to become a 2 carbon. So remember, I still have 2, but I have 2 carbon chains. That carbon is released as a CO2. And I also have some electrons that get released that create some NADH. It's a win-win situation. I have my 2 carbon chain. It no longer can be called pyruvate because it's only 2 carbons. What do I call it instead? Acetyl-CoA. Excellent, acetyl-CoA. Okay, so now these guys are ready to go into the Krebs cycle. But can they go at the same time? No, if they try to, they bump into each other. It's like trying to walk through a door at the same time. You're going to get stuck. So they have to take turns. One of these is going to go through, and when it goes through, it's going to generate 1 ATP. It's going to generate 3 NADHs, and it's going to generate 1 FADH. Okay, now the other one has to go through, and it's going to do the same. It's going to do 1 ATP, 3 NADHs, and 1 FADH2. Right now, FADH2. Okay, so now, what is my net total going to be? Well, I have to add them together. So if I say on a test, what is the output for 1 acetyl-CoA? 1 acetyl-CoA, it would be 1 ATP, 3 NADHs, 1 FADH. If I say, what is the output net at the end of the Krebs cycle, after they've both gone through, I'm going to have a net total of 2 ATP, 6 NADHs, because I'm adding them together, and 2 FADH. Does that make sense now?
It makes a lot more sense.
Makes a lot more sense? Good. Okay, so we're feeling good. Okay, yes, Jen.
So, what did you say the question was? If it's 2.
So if I ask you, what is the net total after an acetyl-CoA goes through the Krebs cycle? That would be those singles. If I ask you, what is the net total at the end of the Krebs cycle? When the Krebs cycle is complete and we're done. At that point, they both have gone through. So that's where I get those increased totals. Okay, does that make sense? Okay, good. Now, the Krebs cycle, it is going to be a process that for us is going to occur in the mitochondria. So it's going to be where we're going to make a little energy in our mitochondria. For prokaryotes, it's going to occur in their cytoplasm. So they're going to do glycolysis in their cytoplasm, and because they don't have a mitochondria, they have adapted to do Krebs in their cytoplasm as well. So everything that they have is just going to stay in the cytoplasm for now. All right, we're special, we have to move to the mitochondria. All right, so our process then, we're going to have our 2-carbon chain enter. And you see that at the top with the arrow. It says acetyl-CoA enters. As soon as acetyl-CoA enters the Krebs cycle is like a big spinning wheel. As soon as that 2-carbon enters, it has a 4-carbon waiting for it. My 2-carbon comes in, my 4-carbon molecule hits it, and now I have a total of a 6-carbon molecule. This is citric acid. This is why it's called the citric acid cycle, because the first thing I make is citric acid. All right, because the 6-carbon, the way it's arranged, is citric acid. Now what I'm going to do, I have a 6-carbon molecule. My goodness, I already started glycolysis with a 6-carbon molecule, and now I have another one. Oh my gosh, it's like I'm going backwards. But I'm really not. I'm going to start rearranging it. So the first thing I'm going to do is I'm going to rearrange that 6-carbon molecule by removing one of the carbons. When I remove a carbon, remember, it gets removed or kicked off as a CO2. Okay, so I remove one of the carbons. I become a 5-carbon. Now, that CO2 goes off. We release it when we respire, when we breathe out. And when I kick that carbon off, I release some hydrogens, which generate some electrons that get grabbed by NAD. Okay, so I broke off some carbons. So now my NAD goes to NADH. I now have a 5-carbon molecule. What am I going to do? Well, one more time, I'm going to kick another carbon off. So I kick another carbon off. I'm going to, again, have some electrons get released. I make some more NADH. I'm going to release that carbon as a CO2. Now, this is where it gets a little odd. What's going to happen here is when I have this 4-carbon molecule, I'm going to rearrange it a little. And when I rearrange it, I generate some energy waves. And when I generate some energy waves, I get enough energy to attach a phosphate onto ADP. That's pretty cool. That's where I get my single ATP. It's because as I start moving stuff around, just like when we're moving around, we're creating energy. I start moving things around and creating a little energy. Okay, so I get a little ATP made right here. I then keep rearranging my molecules. And when I rearrange them, I start doing a double bond instead of a single bond, things like that. And that releases another hydrogen, which means I get an FAD come in and grab that electron. So I get my FADH. I rearrange again. I get another electron release. This one gets grabbed by NAD. It's whichever Uber driver is nearby. Okay? And now when I'm done, I'm back to a 4-carbon molecule. And that carbon goes back in, bumps into the 2-carbon coming in, and the whole process repeats. Okay? Now, again, this is very simplified because I really want you to understand the end result, what we're trying to achieve. Rachel.
So the FAD and the NAD, so it just depends on which one it bumps into at the time, so it doesn't necessarily have to be NAD, like this particular.
Good question. So they each come in different parts of the cycle. So at the very beginning, I would always get NADH because my NAD kind of hangs out at the beginning. And my FAD is kind of hanging out towards the end. They each have like little strategic areas they're hanging out at where they know something's going to happen. And the reason they know that is because the enzymes have put them there and told them, wait here, something's coming your way. So the enzymes are helping make sure that we're in the right locations. Okay? All right. So when we're done with the Krebs cycle, I know this is like mind-blowing. Metabolism day is like, oh, don't worry, we're going to recap next time too. We're going to go through it until we start to look at it. What's that? It's overwhelming? Yeah, it's a lot. It's a lot. I'm going to tell you guys, when I first saw this, I sat there and went, ouch, my head hurt. It like hurt. And I was like, this doesn't make sense at all. It didn't make sense. But do you know what helps? Is to sit there and write it out. Just sit there and draw it out. And I don't mean like drawing all the steps, but the main steps is gonna help. Yeah.
I'm sorry to say this, but I'm gonna be honest to you.
Yeah.
Some people here we did learn by trying.
Yes.
And I got that what you do right now. I mean, sometimes you go kind of fast.
I do.
And,
Yes.
Unfortunately, I would like you to go slower.
Okay, I will
Yeah,
try.
I'm sorry.
I totally appreciate
And,
that feedback. I do.
And sometimes like students will get it, like you will take more time doing what you just did right now over there instead of just going through slides. And some people like to see it and you try and you learn. That's how I learned that right now.
And I appreciate that. And here's what I do. When I stop and draw things, it's because I'm looking at your faces. If I'm seeing 90% of my class is looking confused, we're going to slow down.
I mean, some people will see it like that.
Right. And if I see a lot of people getting it, I'm going to keep going. But here's the deal. If it's still confusing, see me during office hour or see me after lab, and then you and I can go through it slower, step by step. But I kind of base it on how the majority are feeling, because I can tell by your faces. I can look around the room and I see either this or I see this. I can tell. And that's what I'm trying to base it on. And you're right. Some students are going to be like, I need you to draw it. That's fine. Talk to me after lab and I will draw it out. I have no problem with that. And I appreciate that feedback. And you're right too. We do go fast in here because we have a lot of material to cover. That's the unfortunate thing. So we do. We have a lot to cover. But I appreciate that. I love that. It's so good. Okay, so when we go through this then with our glucose, when we're done, so after we've gone through the Krebs cycle, right? We went in with the acetyl-CoA. We walk away with 6 NADHs. That's our total. We walk away with 2 FADH2s. And we walk away with 2 ATPs. All right, so now we're done with the Krebs cycle. At this point in time, we're ready to go on to the main event, which is the electron transport chain. The idea behind this is we've done a lot of work. We've taken glucose, we've basically broken it apart, we have reconstructed it, and we've done a lot of work for very little ATP. I got 2 in glycolysis. I got 2 total in Krebs. I'm exhausted. I'm tired of this. When am I going to actually get some energy? Well, now we're to the electron transport chain, and this is where it all works. All of those steps that we did up to this point was getting ready for the ETC. So having that ATP preparation, meaning that we're putting electrons on NADH, we're putting electrons on FAD, this is where it's going to pay off. Okay, so the electron transport chain, this is going to be where we create the most ATP. So what's going to happen here are those Uber drivers, our NADH and our FADHs are going to arrive in the inner membrane of the mitochondria for us, or if they're prokaryotes, they're going to use the plasma membrane. They just have to have a membrane, okay? Remember, the reason they have to use the plasma membrane if they're prokaryotes is because they don't have a mitochondria, okay? So what's going to happen now is that we are going to have the ability to have these electrons that are being shuttled by our NADHs and our FADHs to actually generate energy to create some ATP. Okay, so this is where we'll get into our electron transport chain. So the first to arrive in the electron transport chain process are the NADHs. Because of this, NADH, for each one that arrives, is going to generate a little more ATP than FADH. It's because of where it arrives in the process. So our NADH is going to arrive to that inner membrane of the mitochondria. And what's going to happen is the electrons that are sitting on the NADH are going to get pulled off of it by an electron acceptor. The way you can think of the electron transport chain is it's like a game of hot potato. You guys ever played hot potato? You get like a pillow or something and you toss it to the next person, and the idea is that you just kind of keep tossing it because you don't want to be the one holding the electrons. That's kind of what's going to happen here. A molecule is going to see NADH, it's going to grab its electrons, and then beside it is a bigger molecule that's going to grab the electrons, and then beside that is another molecule that's going to grab the electrons, and they're going to move down a chain. That's why we call it the ETC, the electron transport chain, because we're pulling electrons. Now, sitting at the very end of our chain is the biggest electron puller. It's our bowling ball on the sheet. Who is it?
Oxygen.
Oxygen. So for us, way at the end of the chain is the oxygen. And oxygen loves electrons and it's going, gimme, 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 gimme. It's pulling those electrons towards it. All right, so the electrons are going from one to another, one molecule to another, but they're going towards that final pull, which is the oxygen. If I take oxygen away, I get stuck. There's no pull. And so if something gets an electron, it's like there's no oxygen at the end and keep pulling. It's like, I don't know what to do with this. Do I just let it go? Like it stops. Everything stops. I need oxygen at the end to keep pulling. Okay, it's gonna pull that chain along. You got a question or you good?
Yeah, I don't know if it would make any sense, but would that make any difference for like in different athletes, like swimmers or anything like that?
So it doesn't affect them as long as they have oxygen present. Right. When you're swimming, you have to breathe, so you still have oxygen present. To compensate, what they have is they have extra mitochondria. So the mitochondria, when they breathe in their oxygen, there's lots of mitochondria that are using it. And so the athletes will have more mitochondria to keep the process going. But that breath of air is enough to keep the process going. Isn't that cool? It's like cool stuff. Okay, so we're gonna drop off our electrons and they're gonna pull down this chain. Okay? Our NADHs are going to arrive first. Now, the other thing about these electrons is remember where they came from. Who donated the electrons to begin with? What molecule? Not carbon.
Hydrogen.
Hydrogen. Thank you. Hydrogen. Hydrogens were the ones who gave their electrons away to begin with. Now remember what I told you. Electrons are negative. If a hydrogen gives them away, it becomes a hydrogen ion. It becomes positive. And it is going to be attracted to the electrons. So we run into a situation. The NADH arrives. It passes its electrons along. But the hydrogens are released. The hydrogen ions, they see those electrons. And have you ever had seller's remorse? Where you gave something away and you wish you still had it? Anybody ever had that? I get that every now and then. Like I'll clean out my closet and I'm like, my closet's clean and I give away a whole bunch of stuff. I'm like, I wish I had that jacket still. Like, it's that kind of like, donator's remorse. Okay, the same is true here. The hydrogens gave up an electron, but now they kind of want it back. They're like, they're positive, they're attracted to it. So what happens is as the electrons are shuffled from one acceptor to the next, the hydrogens follow along trying to get the electrons back. They want to reunite with the electrons. And we let them reunite at the very end. And when they reunite, they generate some ATP. So here's our process. So my NADH comes in, that's in yellow. My FADH comes in, that's in blue. NADH arrives first, so I get a little more energy generated from the NADH just because of where it arrives. Okay. What's going to happen? They drop off their electrons. And their electrons get passed from one acceptor to the next. Those are the things in circles. Those are molecules grabbing electrons and passing them to the next one. Okay? So electron acceptors are in circles. Notice the pink arrows. The pink arrows are showing you that those are the hydrogens that lost their electrons being pumped up and across this membrane so that my electrons are on one side and my hydrogen ions are on the other. There's a membrane separating them. Okay? So I have my hydrogen ions and my electrons. I'm going to have those hydrogen ions keep following on the opposite side of that membrane until they reach a special enzyme called ATP synthase. What does that sound like it makes? ATP synthase, what do you think it makes? ATP, yeah. Remember, enzymes make sense. If I'm synthesizing ATP, I'm ATP synthase. So here's the deal. This enzyme is a special tunnel enzyme. And what it allows is it allows the hydrogen ions to flow through to reunite with their electrons. Oh, it's so good, reuniting, okay? When this happens, when they flow through, it's like a waterfall on a water wheel. It'll spin this enzyme. This enzyme starts to spin, and that creates energy. That spinning or that motion, that energy creation, is going to be enough to attach phosphates onto ADP. Isn't that cool? Okay, we're gonna pick this up here next time and we're gonna go through this all again. I'm gonna keep hitting you with it. All right, we're just gonna keep doing it until it sounds like it makes sense. All right, so go ahead and let's take a break. Since I went over, we'll start lab at 10:35. So that way you still have your full 10 minutes.